Welcome you all in this session 2021 NEET question paper. Botany questions of this question paper is discussed. Botany questions of 2021 NEET examination. First question: mutations in plant cells can be induced by. Mutation can be induced by certain agents. They are called mutagens. Mutations can be divided into two: spontaneous mutation, that is naturally occurring mutation, and induced mutation. Induced mutations can be achieved by the application of mutagens. So mutagen is a substance, or mutagens can induce mutation. There are two types of mutagens: physical mutagen and chemical mutagens. Physical mutagens are radiations. Chemical mutagens are chemical substances. Here the answer is option 3. Option 3, gamma rays. Gamma ray is a physical mutagen. Kinetin and cysteine. Kinetin and cysteine, they are cytokinins, a class of plant hormone, cannot induce mutation. Infrared rays are radiation, but cannot induce mutation. The mutation capable is that is gamma radiation. Then here it is a match the following. Then cells with active cell division capacity, it is meristematic tissues. Tissues having all cells similar in structure and function. They are simple tissues. Then tissues having different types of cells, they are vascular tissues, because vascular tissues have different types of components. For example, xylem has 4 components: tracheids, vessels, xylem parenchyma, xylem fiber. Phloem has 4 types: sieve tube element, companion cell, phloem parenchyma, phloem fiber. So both xylem and phloem, they constitute the vascular tissues, consist of different types of elements, different types of cells. Then dead cells with a highly thickened walls and narrow lumen, it is sclerids. So the answer is A. So the answer is A. A2, B4, C1 and D3. Then next is match the following. Lenticels can be matched with 3, because they are aerating pores and exchange of gases. Cork cambium can be matched with 1 because phellogen is the other name of cork cambium. Secondary cortex can be matched with 4 because the other name is phelloderm. Cork, it is matched with a 2 because cork cells have suberin deposition. Answer is 2. Answer is 2. Answer is 2. Next question. Complete the flowchart of central dogma. Complete the flowchart of central dogma. Then in the flowchart, 4 items are asked to identify A, B, C, D. A is DNA to DNA. It is replication. A is DNA to DNA replication. B is DNA to mRNA transcription. C is mRNA to dash, mRNA to protein. So C is translation. D is protein. That is, answer is 3. Replication, transcription, translation, and protein. This is what is called the central dogma of molecular biology. Central dogma of molecular biology. It was proposed by Francis Crick. Then, next, the term used for transfer of pollen grains from anther of one plant to stigma of a different plant, which during pollination brings genetically different types of pollen grains to stigma. This type of pollination is known as synogamy. There are 3 types of pollination: autogamy, geitonogamy, and synogamy. Autogamy is the transfer of pollen grains from the anther to stigma of the same flower. Anther to stigma of the same flower. Geitonogamy, transfer of pollen grain from anther to stigma of different flower but of the same plant. Transfer of pollen grains from anther to stigma of different flower, but the flowers are on the same plant. Geitonogamy is functionally a cross-pollination, seems to be a cross-pollination, but genetically it is a self-pollination because both the plants are on the same plant. Synogamy is the true cross-pollination. Synogamy is the true cross-pollination. Here, that is transfer of pollen grain from anther to stigma of another plant situated on another flower of another plant. Synogamy is the true cross-pollination which brings about genetic variation. Then, next is next question. Then it is a question number 108 of botany part. Which of the following is an incorrect statement? Which of the following is an incorrect statement? First statement is incorrect. First statement is incorrect. Mature sieve tube elements possess a conspicuous nucleus and usual cytoplasmic organelles. It is wrong. Mature sieve tube elements does not possess a conspicuous nucleus. So the statement is wrong. Answer is option 1. Then the next question of botany is gemme are present in some liverworts. Gemme are present in some liverworts. What are gemme? Gemme are asexual reproduction structures for asexual reproduction. Structures for gemme are structures of asexual reproduction. They are multicellular, green colored structures. They have a stalk. They are produced in small cavities called gemme cup. And these gemme are present not in all liverworts, but in some liverworts. The example is Markantia. Markantia is a liverwort which possesses gemme and gemme cup. Next. Next botany question. Question number 111. So protoplast fusion can be matched with 2 because tomato is a plant produced by the protoplast fusion of potato and tomato. Protoplast fusion. This is what is called somatic hybridization. Then plant tissue culture can be matched with 1 because plant tissue culture is based on the principle, or it works on the principle of totipotency. Meristem culture can be used with matched with 4 because explant used for producing virus-free plant, they are meristem. Meristems are free from viruses. Micropropagation, it can be matched with the somaclodes. So during micropropagation, large number of progeny are produced through tissue culture and they are genetically similar and they are called soma clones. Answer is 2. Next botany question is 112. Production of gametes by the parents, formation of zygote, F1 and F2 plants can be understood from the diagram called, which we use to represent formation of zygote, F1, F2, etc. It is, the diagram is called Punnett square. It was first developed by R.C. Punnett to show the gamete, fusion of gamete, F1, F2 generation, etc. Then next question is 113. Genera like Selaginella and Salvinia produce two types of spores. Such plants are called heterosporous plants. What is heterospory? Formation of two types of spores is called heterospory. Such plants are called heterosporous plants. Two types of spores are referred as microspore and megaspore. Microspore and megaspore. Microspores are small and they are the male spores. Megaspores are large and they represent the female spore. Next question of botany is question number 16. 116. Question number 116. It is a match the following. It is a match the following. First is cohesion. Cohesion. It can be matched with two. Attraction, mutual attraction among water molecule. Next is adhesion. It can be matched with 4. Attraction towards polar surface of tertiary element. That means attraction between water molecules. Attraction between water molecules and the wall of tertiary element. It is called the adhesion. Then surface tension. It is more in liquid phase. Guttation, water loss in liquid phase. Answer is 1. Water loss in liquid phase. Answer is 1. Then next question of botany is 120. 120. First stable product of carbon dioxide fixation in sorghum. Sorghum is or maize is a C4 plant. Sorghum or maize is a C4 plant. In C4 plant, the first stable product is oxaloacetic acid, a 4-carbon compound. Oxaloacetic acid, a 4-carbon compound. How it is formed? Phosphoenol pyruvic acid combines with the carbon dioxide. The enzyme is PEP carboxylase. The enzyme is PEP carboxylase and product is oxaloacetic acid. PP is a 3-carbon compound. Carbon dioxide is a 1-carbon compound. Oxaloacetic acid is a 4-carbon compound. The first stable product of C4 pathway is oxaloacetic acid. It is a 4-carbon compound, so it is called C4 pathway. C4 plants show C4 pathway. Then next question. Which of the following algae produce carrageen? Carrageen is a hydrocolloid. Which of the following algae produce carrageen? Carrageen is produced by red algae by name chondres. This algae produces carrageen. Carrageen is a colloidal substance. It is a hydrocolloid or can be called a phycocolloid. Next question 121, 123, 123. Then match the following. Cristae can be matched with 3 because they are enfoldings of the inner mitochondrial membrane. Thylakoid can be matched with 4 because they are flattened membranous sacs found in the stroma of a plastid. Centromere is the primary constriction. Centromere is primary constriction in chromosome, and cisternae, they are disc-shaped sacs in Golgi apparatus. So the answer is 3. A3, B4, C1 and D2. Then next question, 124. Which of the following are not secondary metabolites? The answer is amino acids and glucose. What are secondary metabolites? Secondary metabolites are chemicals or molecules having no role in the physiology of the plant. Secondary metabolites have no role in the physiology of the plant. So they are, but primary metabolites, on the other hand, they have a role in the physiology of the plant. Amino acids and glucose, they are primary metabolite. Others are secondary metabolite. Answer is 2. Next, which of the following algae contains mannitol as reserve food materials? That is, mannitol is found as a reserve food material in Phaeophyceae. Mannitol is found as a reserve food material in Phaeophyceae. We have to select a Phaeophyceae member. Ectocarpus is a Phaeophyceae member. Gracilaria is a Rhodophyceae. Volvox and Eulothrix are Chlorophyceae. Volvox and Eulothrix are Chlorophyceae. Gracilaria is Rhodophyceae. So the answer is option 1, Ectocarpus. Mannitol is a reserve food materials which is found in Phaeophyceae brown algae. Others, this one is red algae and these two are green algae. Next is question number 106. Typical angiosperm embryo sac at maturity. It is an 8-nucleate 7-celled structure. 8-nucleate and 7-celled structure. Angiosperm, female gametophyte of angiosperm is also called embryo sac. It consists of 7 cells but 8 nucleus. The difference in the cell number and nucleus number, it is because one cell in the embryo sac of angiosperm, that is central cell, consists of 2 nuclei. These nuclei are called polar nuclei. These nuclei are called polar nuclei. Then Next, diadelphous stamens. 127. Diadelphous stamens are found in. Diadelphous stamens are found in P. What is adelphi or adelphous condition? Filaments of the stamen are fused, but anthers are free. Filaments of the stamen are fused, but anthers are free. If the filaments are fused into one bundle, it is called monoadelphous condition. It is called monoadelphous condition. If the filaments are fused into two bundles, it is called diadelphous condition. If the filaments are fused into more than two bundles, it is called polyadelphous condition. Polyadelphous condition. Monoadelphous condition. Example, hibiscus. Diadelphous condition. Example, P. Polyadelphous condition. Example, citrus. China rose is monoadelphous. Citrus is polyadelphous. P is diadelphous. Then option 3 the answer. Next botany question. Question number 129. Which of the following stages of meiosis involves division of centromere? Centromere splitting. Centromere splitting takes place in which of the following phase? Centromere splitting takes place in the anaphase 2. Centromere splitting in meiosis takes place in anaphase 2. Centromere splitting in mitosis takes place in anaphase. During anaphase, the centromere split and the chromatids are separated. Centromere split and the chromatids are separated. So it occurs during anaphase 2 in meiosis, anaphase in mitosis. Answer is 3. But anaphase 1, in anaphase 1 of mitosis, there is no centromere splitting. Anaphase 1 of mitosis, there is no centromere splitting. The homologous chromosomes separate. Homologous chromosomes separate and they move to the opposite pole. There is no centromere splitting. In anaphase 1, there is no centromere splitting. Chromosome move towards the homologous chromosomes separate and they move towards the opposite poles. Anaphase 1, there is no centromere splitting. Anaphase 2, there is centromere splitting. Then, next question. Plants show different pathways in response to environment or phases of life to form different kinds of structure. The phenomenon is known as plasticity. That is, plant development is flexible. Plant development is in response to the environmental influence, or plant development is influenced by the environment. Depending upon the environmental changes, different types of development takes place in plant. This is what is called the plasticity. Plasticity. Next. Which of the following plant is monoecious? Which of the following plant is monoecious? Monoecious means unisexual. Monoecious plant means bisexual. Monoecious plant means bisexual. That is, m- male and female reproductive organs are found in one and the same plant. One and the same plant. Monoecious means bisexual plants. Answer is Kara. In Kara, male reproductive organ and female reproductive organ are found in the same plant. Male and female reproductive organs are found on the same plant. 132. 132. When the centromere is situated in the middle of two equal arms of a chromosome. When the centromere is situated in the middle of each two equal arms of chromosome, the chromosome is called a metacentric chromosome. Chromosomes are divided into 4 types based on the position of centromere. If the centromere is exactly in the middle, it is called metacentric chromosome. If the centromere is little away from the center, it is called submetacentric. If the centromere is towards one end, that means towards one end, not at the end. If the centromere is towards one end of the chromosome, that is called acrocentric. In acrocentric chromosome, one chromosome will be too small and one chromosome will be large. One chromosome will be large. In submetacentric, the centromere is situated little away from the center. Then in acrocentric chromosome, centromere is situated towards one end of the chromosome. So one end will be much longer than the other. Long arm is called Q arm. Short arm is called P arm. Long arm is called Q arm. Short arm is called Q P arm. P short arm, Q long arm. Next, telocentric chromosome. If the centromere is present at the tip of the chromosome, centromere is present at the tip of the chromosome is called telocentric. Tip of the chromosome is called telomere. Tip of the chromosome is called telomere. Next. Site of perception of light in plants during photorespiration. What is photorespiration? Photorespiration is a phenomena in which plants show response to light in flowering. This is what is called photoperiodism. Response of plants in flowering towards light is called photoperiodism. So this light, that is the external stimulus, is accepted by which organ? Accepted by which organ? That is leaf. The organ for perception of photoperiodic stimulus is leaf. Next is 134. The factor that leads to founder effect in a population, it is called genetic drift. Genetic drift. What is meant by founder effect? Founder effect is a genetical phenomena in which genetical phenomena in which brings about genetic drift. What is founder effect? That is when a small group of individuals from a population migrated into another. That is a small group of population. That is, this is a population, suppose. This is a population. This is another population. From the first population, that is a small group of individuals, they migrated from this population to a new population. Small group of individuals, they migrated from a population to a new population. New population is also, this is a population. A small group of individuals migrated from a population. This group, now it is called founders. A small group of individuals from a population migrated into a new population. The group of individuals which migrated into the new population, this group is called, now this group is called the founders. These founders, they with sexual reproduction with the other members of the population, they brings about a variation in this population. Brings about variation in this population. This variation, it is called genetic drift. So founder effect is a cause for genetic drift. Founder effect is a cause for genetic drift. Next is 135. The plant hormone used to destroy weeds. It is 2,4-D. 2,4-D, a synthetic auxin. 2,4-D is a synthetic auxin used as a weedicide. It is a synthetic auxin. It is used as a weedicide. Then, next, match the following. So, nitrococcus can be matched to 2. Nitrococcus can be matched to 2 because they involve in the conversion of ammonia to nitrate. Conversion of ammonia to nitrate. Ammonia to nitrite. Conversion of ammonia to nitrate. Conversion of ammonia to nitrate is called nitrification. Conversion of ammonia to nitrate is called nitrification. In the first step, ammonia is converted to nitrite. In the second stage, ammonia is converted to nitrate. So conversion of ammonia to nitrate, conversion of ammonia to nitrate is called nitrification. It occurs in 2 stages. In the first stage, ammonia is converted to nitrite. 2 bacteria are involved, Nitrosomonas and Nitrococcus. This step, Nitrosomonas and Nitrococcus. Nitrosomonas is not given, Nitrococcus is given. So Nitrococcus involved in this first step. Rhizobium, it is second B. It is rhizobium. it fixes atmospheric nitrogen. That is, conversion of atmospheric nitrogen to ammonia is called nitrogen fixation. Conversion of atmospheric nitrogen to ammonia is called nitrogen fixation. So rhizobium involves in. Then thiobacillus, it is denitrification. Nitrobacter, conversion of nitrite to nitrate. Conversion, second part of nitrification. Conversion of nitrite to nitrate. Conversion of nitrite to nitrate. So the answer is option 1, 2, A2, B4, C1 and D3. Next, select the correct pair. Select the correct pair. First pair, large colorless empty cells in the epidermis of grass leaf. It is not subsidiary cells. They are bulliform cells. Bulliform cells. Subsidiary cells are cells surrounding the guard cell. Subsidiary cells are cells surrounding the guard cells. Then, second one, in dicot leaves, vascular bundles are surrounded by large, thick-walled cells. It is not conjunctive tissue. They are bundle sheath cells. They are bundle sheath cells. Conjunctive tissue. Conjunctive tissue, they are parenchyma cells present in between xylem and phloem. In between xylem and phloem of root. Xylem and phloem of root. Xylem and phloem of root. Then, next question is 138. It is 139. Botany part 139. Identify the correct statement. Then, correct statement. First statement. Then, in capping methyl guanosine triphosphate is added at the 3' end is wrong. It is added at the 5' end. That is after transcription in eukaryote, especially the hnRNA undergo 3 post-transcriptional changes. hnRNA is modified, undergoes some processing. This processing is known as post-transcriptional changes. One such a post-transcriptional change is capping. Capping is the methyl guanosine triphosphate is added at the 5' end. Then, second one, RNA polymerase binds with the rho factor to terminate the process of transcription in bacteria. Correct. Rho factor is the termination factor. Rho factor is the termination factor. Sigma factor is the initiation factor. During transcription, to initiate transcription, RNA polymerase binds with the sigma factor. RNA polymerase binds with the sigma factor. To stop transcription, RNA polymerase binds with a rho factor. Rho factor. So that statement is correct. We want a correct statement. Correct. Then the coding strand in transcription unit is copied to mRNA. Wrong. It is the template strand. During transcription, template strand is copied, not coding strand. Template strand is copied and it is not the coding strand. Template strand is copied. It is not the coding. Nothing is formed from the coding strand. But template strand, it is copied into RNA. So RNA will be complementary to the template strand. Nothing is coded from, nothing is produced from the coding strand. Then split gene arrangement. Split gene arrangement is the characteristic of prokaryote. Wrong. Split gene arrangement is the characteristics of eukaryotes. Split gene arrangement is the characteristic of eukaryotes. What are split gene? Split gene is a gene which consists of exons and introns. Split gene is a gene which consists of exon and intron. What are exons? Exons are coding sequences. Exons are coding sequences and they are present in the mature mRNA. Exons are coding sequences and they are present in the mature mRNA. Introns are non-coding sequences and they are absent in mRNA. So that is why it is this type of gene. Split genes are not in prokaryote but in eukaryote. That means eukaryotic gene consists of both exons and introns. Eukaryotic gene consists of both exon and introns. But prokaryotic gene consists of only exons. Prokaryotic gene consists of only exon. That means introns are absent in prokaryotic genes. The exponential growth equation NT equal to N0 into e to the power of RT. Exponential growth equation means geometric type growth. In geometric growth, the equation, the growth curve, that is sigmoid curve, exponential growth it is called, it is represented by this equation. Here E represent the base of natural logarithm. It is 2.7 something. It is a constant. Next, 141. Which of the following statement is incorrect? Which of the following statement is incorrect? Statement number 2 is incorrect because in ETC, one molecule of NADH plus H plus gives rise to 3 ATP molecule, and one FADH2 gives rise to 2 ATP molecule. So that is wrong. NADH plus H plus can give rise to 3 ATP. FADH2 give rise to, that is 2 ATP. FADH2 gives rise to 2 ATP. Then which is the role of RNA polymerase 3? In eukaryotes, there are 3 types of RNA polymerases, at least, or we can say some 3 or 4 types of polymerase. Mainly, there are 3 types: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 3. Apart from these, eukaryotes also possess, that is RNA polymerase for chloroplast and mitochondrial RNA. However, 3 RNA are there, that is RNA polymerase 3. RNA polymerase 3 transcribes tRNA, 5S rRNA, snRNA, 5S rRNA and snRNA. Then first one, this is RNA polymerase 1. Then mRNA, it is transcribed by RNA polymerase 2. RNA polymerase 2. Then snRNA, it is polymerase. It is not only snRNA. That is why we dropped. RNA polymerase 3 transcribes not only snRNA, also tRNA and 5S RNA. 5S RNA. So the only we dropping the RNA polymerase 2, RNA polymerase 3, they transcribe 5S rRNA, tRNA and snRNA and snRNA. Then in some members of which of the following pairs of family pollen grains retain their viability for months after release? A question related to pollen viability. What is pollen viability? That is, after the release of pollen grain from the anther, for how much duration the pollen grains can retain the power of effective pollination. How long the pollen grains retain the power of effective pollination. That is what is called pollen viability. In some plants, for example in poaceae, it is just 30 minutes. Pollen viability is 30 minutes. That means after the release of pollen grain from the anther, if pollination could not occur Before 30 minutes, the pollen grains become non-viable. That means it could not bring about an effective pollination. The best example is Poaceae family. In Poaceae family, the pollen viability is very short. But in some families, for example, Rosaceae and Leguminosae, the pollen viability period is, for example, for months, for 2 or 3 months, the pollen viability retain. So the answer here is Rosaceae and Leguminosae. That is viability for months after their release. Months. Then next, match the following. Choose the correct option. That is, S phase can be matched with the 4 because DNA replication takes place there. G2 phase, proteins are synthesized. Quiescent stage is an inactive stage. G1 phase, interval between mitosis and initiation of a replication. That is the period between one mitosis and the next DNA replication is called the G1 phase. G1 phase. So answer is 4, 1, 2, 3. 4, 1, 2, 3. Answer option 3. Answer option 3. 4, 1, 2, 3. A4, B1, C2, D3. Then there is no botany question there. In 45, next page it is. Match the following column 1 and column 2. So here floral formula is given in column 1 and family name is given in column 2. First floral diagram. It is that of Fabaceae. First floral diagram, because zygomorphic, bisexual, K5, polysepalous, C1 plus 2 plus 2. The third component is fused. Then A9 fused plus 1. Gynoecium, ovary superior, monocarpellary. It is that of Fabaceae. Then second one, B, it is the floral diagram is that of Solanaceae. Regular, actinomorphic, bisexual, K5, gamosepalous, C5, gamopetalous, A, stamens 5, epipetalous, gynoecium, ovary superior, bicarpellary, syncarpous. So next it is that of a Liliaceae. Here perianth is present. There is no distinction between corolla and calyx. So we identify it as perianth. Actinomorphic, bisexual, perianth 3 plus 3, gamotepalous, andrecium 3 plus 3, epitepalous, gynoecium, ovary superior, tricarpellary, syncarpous. Tricarpillary syncarpus. It is D is that of a Brassicaceae. Here, actinomorphic, bisexual, K2 plus 2, corolla 4, A2, then 2 to 4, G 2 plus 4 it is actually, 2 plus 4, then G ovary superior, bicarpillary. Answer is 1. Floral formula and correct match of the family. Then next question, 148. Which of the following statement is incorrect? Which of the following statement is incorrect? Among the given, the fourth statement is incorrect. Cyclic photophosphorylation involves both PS1 and PS2 is wrong. Cyclic photophosphorylation involves only PS1. Cyclic phosphorylation or cyclic photophosphorylation involves only PS1. Only PS1. Then PS1 and PS2, non-cyclic photophosphorylation. Non-cyclic photophosphorylation involves PS1 and 2. PS1 and 2. PS1 and non-cyclic photophosphorylation occurs in the grana lamellae. Non-cyclic occurs in the grana lamellae. There you can see PS1 and PS2. But cyclic photophosphorylation occurs in the stroma lamellae. Occurs in the stroma lamellae, and there is only the involvement of PS1. Then next page, one question is from the botany side. Then question number 149. Proteins can be matched with the item number 4 of list 2 because proteins show polypeptide. Proteins contain polypeptides. Polypeptides. They are polypeptides. They have peptide bond. What is peptide bond? Bond formed between the carboxyl group of one amino acid and amino group of another amino acid. Then unsaturated fatty acid. Unsaturated fatty acid. Unsaturated fatty acid can be matched with the item number 2 because they have double bonds. Unsaturated fatty acids have double bonds, but saturated fatty acid have only single bond. Saturated fatty acids have only single bond. Palmitic acid is a saturated fatty acid. Arachidonic acid is an unsaturated fatty acid. So unsaturated fatty acids have double bond. Saturated fatty acid have only single bond. Nucleic acid can be matched with item number 2 because nucleic acids are polynucleotides. Nucleic acids are polynucleotide. Polynucleotide. That is, consist of many nucleotides. These nucleotides are linked together by phosphodiester bonds. Nucleotides are linked together by phosphodiester bonds. Then D can be matched with 3. That is polysaccharides. They possess glycosidic bond. What are glycosidic bonds? Glycosidic bonds are formed between two adjacent monosaccharides. Two adjacent monosaccharides into adjacent monosaccharides in a polysaccharide. Then the two adjacent monosaccharides in a polysaccharide. Then next botany question is in the next page. Organelles that are included in the endomembrane system. Organelles that are included in the endomembrane system. What are endomembrane system? Membrane bound organelles having a coordinated function. Membrane bound organelles having a coordinated function. Membrane bound organelles having a coordinated function, they are called endomembrane system. 4 cell organelles are included in the endomembrane system. They are endoplasmic reticulum, Golgi endoplasmic reticulum, Golgi complex, lysosomes and vacuole. So endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Endomembrane system is membrane bound. We can say single membrane bound cell organelles, and they have a coordinated function. That means their functions are closely related or interconnected. Such organelles are called, collectively called, endomembrane system. 4 cell organelles are there in endomembrane system. Endoplasmic reticulum, Golgi complex, lysosome and vacuole. Next, that is next botany question is, in a cross between male and female heterozygous, male and female are heterozygous for sickle cell anemia. Sickle cell anemia. Then sickle cell anemia is due to a recessive allele, that is HBS. That is HBA. It is the normal allele. HBS is the normal allele. It is the dominant allele. HBS is the mutated allele. That is the recessive allele. Recessive allele causes the disorder. If recessive allele causes the disorder, homozygous recessive condition only produce the disease. Only the homozygous recessive condition produces the disorder. That is HBA. This is homozygous homozygous dominant. Homozygous dominant. They are normal. HBA, HBA, HBS. HBS is the mutated allele. They are heterozygous. They are heterozygous, also called carrier. They are normal. They have normal RBC, but called carriers. They have normal RBC and they are called carriers because they carry the recessive allele. On the other hand, HBS, they are homozygous, recessive. They show the disorder. Homozygous, recessive, and they show the disorder. This is the genetic allele. This is the alleles in the sickle cell anemia. This is the genotype of an individual which show that. Then the question is, if father is heterozygous, HBA, HBS, mother is also heterozygous, both of them. The question says that if male and female, that is father and male and female, both are heterozygous, what is the chance of sickle cell anemia, or what is the percentage of sickle cell anemia for their children? We can make use of a checkerboard. In an earlier question we had seen, we can make use of a checkerboard to show the gametic fusion. HBA gamete, HBA gamete. Other, that is HBS gamete, HBS gamete. If this gamete fuses with this one, the individual is HBA, normal. If this gamete is fuses with this, it is HBA, HBS, again normal. Here HBA, HBS also normal. Then this one will be HBS, sickle cell anemia. Sickle cell anemia. So what is the percentage? 3/4 normal, that is 3/4 normal, and 1/4 affected. Affected means affected with the sickle cell anemia. Affected with sickle cell. 1/4 affected with the sickle cell anemia. That means 25%. 25%. So the answer we come to, that is from the option, the answer is 25%. Answer is 25%. Next question comes in the next page, that is match the following. Aspergillus niger can be matched with the citric acid, Acetobacter acetic acid, Clostridium butylicum butyric acid, Lactobacillus or lactic acid. Here it is, some microbes are given which are used for industrial production of organic acid. Answer is 1, A3B1C4D2. Then next is a question that is of botany 1. It is 160. Which of the following is not an objective of biofortification? Biofortification is a plant breeding program in which nutritional quality of the crop is improved. So what is not an objective? Not an objective is biofortification does not aim this. This is correct. This is correct. This is correct. Biofortification is an attempt of plant breeding which aims to improve the protein content, vitamin content, micronutrient and mineral content. But this disease resistance is not coming under the program of biofortification. Next page, that is Dobson, 163. The centriole undergo splits duplication during. Centriole is a cell organelle found in animals. Then centriole undergo replication during S phase. So S phase, two things replicate. One is centriole, other is DNA. Centriole and DNA. Then 164, Dobson unit used to measure thickness of ozone layer. Dobson unit used to for the thickness of the ozone layer. Next 2 pages does not contain botany question. 170, next page, botany question. That is 173. If a fruit fly has 8 chromosomes in the interphase of mitosis, if the number of chromosomes at G1 phase is 8, what will be the number of chromosomes at S phase? During mitosis, chromosome number remains same during all the stages. In all the stages, chromosome number remains the same. Whatever may be. In S phase, DNA replicate, DNA content get doubled, but chromosome number remains same. So answer is 8. Answer is 8. Next question of botany side is this one. If adenine makes 30% of the DNA molecule, what will be the percentage of thymine and guanine? If adenine is 30%, thymine is also 30%. The remaining 40, guanine 20%, cytosine 20%. If the percentage of adenine, what is the percentage of thymine and guanine? Thymine, guanine and cytosine. Thymine is 30%, guanine 20, cytosine 20. Answer is 3, thymine. Thymine 30%. Then that is the botany question. Is next is 178. Which one is 178? Which one is incorrect? Toxin is incorrect. Here alkaloid is codeine correct, toxin heparin correct, lectin concanavanil A correct. Drug resin is not a drug. Resin is a toxin. It is a toxin. So answer is 4. We want an incorrect pair. Drug resin. Drug resin is a toxin. Resin is a toxin. Then 178, 179. Which stage of meiotic prophase? Which stage of meiotic prophase shows terminalization of chiasmata? Then answer 3, diakinesis. Then terminalization of chiasmata, diakinesis. Diakinesis. Then next question is which of the RNA is not required for synthesis of protein? This one, they all required for protein synthesis. They are required for protein synthesis, mRNA, tRNA. Then next, which is the only enzyme that has the capability to catalyze initiation, elongation, termination? It is DNA dependent RNA polymerase. During transcription, RNA polymerase can do all these things. That is initiation, elongation and termination. All these 3 activities can be done by RNA polymerase. Then 186. Which one of the following is not a step in MOET? This is not a step in MOET. It is not H, it is LH. That is FSH-like activity. Having FSH-like activity for superovulation. Others are correct. Then next is 189. Which of the following statement with reference to lipid? That is, choose the correct answer. Which of the following statement? Choose the correct answer from the option. Correct answer from the lipids having only single bond, it is wrong. It is saturated fatty acid, not unsaturated. Lecithin is a phospholipid. Trihydroxypropane is glycerol. Palmitic acid is a 16 carbon compound. Palmitic acid is a 16 carbon compound, not 20. Lipids having only single bond are saturated. Answer is A and this is also wrong. Arachidonic acid is a 20 carbon atom. A, D, what it is? Oh, palmitic acid is 20 carbon atom. Wrong. Palmitic acid 16. Arachidonic acid is D and E. B, C. Oh, correct one. The correct one is B and C. Answer is 3. Answer is 3. B and C only. A is wrong. D is wrong. A is wrong. D is wrong. E is wrong. B and C are only the correct one. Then 190. Which of the following statement about histone is wrong? This is wrong. pH of histone is slightly basic. pH of, so histones are basic proteins. Histones are basic proteins. Histones are basic proteins. 191. 191. The answer is 1. A. Adaptive radiation. It is example Darwin's finches. Convergent evolution. That is third one. Wings of butterfly and bird. Divergent evolution. Bones of forelimb in man and whale. Evolution by anthropogenic action. Selection of resistant varieties due to excessive use of herbicides and pesticides. It is option 1. Then that is in that is botany. It is 195. 95. Statement 1. 2 statements are given. Codon of AUG codes for methionine and phenylalanine. It is wrong for this. Statement 1 is wrong. AUG codes only for methionine. AUG code only for methionine. Statement 2. AAA, AAG, both codons codes for the amino acid lysine. Correct. Statement 2 is correct, but statement 1 is wrong. Lysine has 6 codons, so answer 1 is correct, 1 is incorrect, 2 is true, correct. Answer is statement 1 is incorrect, but statement 2 is correct. So here the botany part question is over. Maybe it consists of some 45 plus or 50 questions. Thank you, dear.